Welcome to the last video of this collection, where our sole focus will be the actin-binding proteins, or ABPs for short. In contrast to the slow turnover of filaments of purified actin at steady state, actin filaments in live cells can polymerize and depolymerize rapidly. This requires the control of more than 60 families of actin-binding proteins. Broadly, these proteins fall into families that bind monomers, sever filaments, cap filament ends, nucleate filaments, promote polymerization, crosslink filaments, stabilize filaments, or move along filaments. Let's zoom in and meet some of the stars among these ABPs and their roles. First, in the spotlight is profilin, a protein that takes center stage by attaching with G-actin subunits, accelerating the formation of filaments. This essential performer plays a pivotal role in constructing the cellular cytoskeleton, the architectural backbone of a cell. Then, we have cofilin, an extraordinary artist specializing in actin depolymerization. With finesse, cofilin attaches itself to the minus end of an actin filament, enhancing the process of filament shortening. By promoting the disassembly of actin filaments, cofilin allows for dynamic rearrangements of the cytoskeleton. Now, let's meet tropomyosin, the stabilizer and regulator of actin filaments, particularly in the realm of muscle contraction. This vigilant gatekeeper controls the interaction between actin and myosin, the powerhouse motor protein responsible for generating the forces behind muscle contractions. Stepping into the scene is thymosin, a captivating actin-binding protein that captivates G-actin subunits, preventing them from binding to existing actin filaments. In this way, thymosin curates the cast, carefully selecting which G-actin subunits get to take part in the actin filament production. A remarkable character known as fermin now takes the stage. This facilitator excels at promoting filament formation and stays devotedly attached to the growing end of the filament, guiding its elongation with precision and finesse. Enter the ARP complex, a captivating ensemble of actin-related proteins. These performers skillfully remain connected to the minus end of actin filaments, allowing for the creation of intricate lattice works by facilitating branching and interconnecting of actin filaments. Their choreographed movements create an awe-inspiring spectacle of cellular architecture. Gelzolin, that slices actin, makes a striking appearance. This artist expertly cleaves filaments and remains gracefully bound to the plus end, leaving behind a captivating trail of actin fragments. The stage wouldn't be complete without the presence of capping protein, a master of stability. This ABP works diligently to maintain the filament's length by skillfully preventing the addition or removal of G-actin subunits, ensuring a seamless and controlled actin performance. And let us not forget the dynamic duo of fimbrin and alpha-actinin, a formidable pair that assists in the formation of actin filament bundles. With their coordinated efforts, they create breathtaking structures that add flair and finesse to the cellular show. In conclusion, actin and actin-binding proteins are the dynamic duo that governs cellular dynamics. Actin provides the structural foundation, while actin-binding proteins finely tune its behavior, regulating processes like cell migration, endocytosis, exocytosis, and cell division. Thank you for joining us on this journey exploring actin and actin-binding proteins. Remember, when it comes to cellular dynamics, actin and its partners are the true stars of the show. For more knowledge, keep watching sqadia.com.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.